So we were just talking about PCR, and prior to that, we were talking about molecular cloning. We were talking about using a piece of DNA and inserting it into a vector for protein expression. Now, with those two things can be kind of paired up. So PCR serves as a great foundation for amplifying a gene that is very, very difficult to find or very obscure or something of that nature. With that PCR product, you can clone that into a vector. Now, those vectors can have characteristics that make them good candidates for purifying a protein. So you have this amplified piece of DNA, you put it into a vector, and those vectors can have things known as basically tags. You make what is known as a fusion protein. Now, these are all examples of good tags for proteins. For instance, let's look at this one right here, the maltose binding protein, also known as MBP. Now, this is a protein that has, well, it's capable of binding the sugar maltose. It's a disaccharide. It's going to bind to maltose. Now, your protein that you're studying, maybe what it does is it catalyzes some sort of oxidation reduction reaction. Getting the product or getting the reactant for that enzyme might be fairly difficult. But if you're able to make a fusion protein between your protein, which I'm going to label YP, and the maltose binding protein, purifying your protein is going to be considerably easier because all that you have to do is immobilize maltose, and all of a sudden now you can capture your protein of interest. So what that actually kind of looks like is this right here. So this is a display of the gene for your target protein and the gene for glutathione S transferase. So those two genes are added together in a vector to make a fusion protein. That fusion protein is expressed within a cell. Those cells are burst open, and the soluble protein fraction is loaded onto a chromatography column. Now, glutathione S transferase is an enzyme that will bind to glutathione. So it will basically bind onto glutathione and stick there until it has a reason to leave. And it will just stay on that column as long as there's still glutathione there. Now, what you can do then is run a solution over this chromatography column and collect any of the proteins that do not adhere to that column. So those proteins that are the proteins that have no affinity for glutathione, well, they're going to flow straight through because your column has glutathione immobilized on it. Now, your protein, which is a glutathione fusion protein, is going to stick to it. It's going to stay there until you add a solution with free glutathione. And that free glutathione, that small molecule glutathione, is going to say, hey, glutathione S transferase fusion protein, go ahead, let's go through this column. And so what you've done is all of these proteins right here, they're junk. They do not have the ability to bind glutathione. Your glutathione fusion protein, on the other hand, well, that's going to stay on the column until you get free glutathione. Now, that's great. That's one way that you can benefit from a fusion protein, by helping purify that protein. On the contrary, other ways that you can use a fusion protein is you can do cellular localization studies. For instance, the green fluorescent protein. You can make a GFP / your protein fusion, and then you can have the ability to actually visualize where your protein is going to be found. So you can look at those cells under a microscope and use a fluorescent light source to see where the green fluorescent protein is expressed, and wherever the green fluorescent protein is expressed, because it's a fusion with your protein, that means your protein is expressed there. Now, that's one way that you can visualize where a protein is. The alternative is you can use a technique known as immunofluorescence, where you tag your protein with a primary antibody and then detect it with a secondary antibody that has a fluorescent tag. So they're both ways that you can basically use a fluorescent label to track down your protein of interest. Now, this immunofluorescence doesn't have the same, I guess, directness to it, because in this case, wherever your protein is expressed, or wherever GFP is expressed, that's where your protein is expressed. Well, that wraps up this chapter, and I'll see you in the next chapter.